Okay, good afternoon, everyone.
Good afternoon.
Okay, Parwan, are we going to start it or not? Yeah.
5 minutes. 5 minutes.
Okay.
5 minutes.
In here we have Dr. Lee Dan from NUS from National University of Singapore. Good afternoon, Dr. Dan.
Hi, good afternoon.
Hello,
Hello.
Dr. Good afternoon.
Pak Cahyo also say hello to you, Dr. Dan.
Yeah. Hi.
How are
Nice
you?
to see you.
Okay,
Yeah.
nice to see you here. Okay, we wait for another 5 minutes to start, Doctor.
Yeah, sure.
Oh,
Shall
yeah.
I share my slides first?
Sure.
Yes, yes.
Mm.
Okay, it's already 1 p.m. in the afternoon. Doctor, what should I call you, Dr. Dan Lee or
My name is Lee Dan.
Ba-
Family name is Lee.
Oh, I see. So your name is Dan, right?
Yes.
Okay. So, Dr. Dan, now the participants is only 54, around 54 people, but I think because the time is already show about 1 p.m., maybe you can start your presentation.
Mm-hmm.
Mm-hmm.
Yeah, sure. Any
Okay.
so you will be letting people in, right? If you...
I will. I just be your moderator because I'm not your host.
Yeah.
The host is the other people, but I'm just delivering. If anyone want to ask to you, then I will be your guy to deliver the
No
question.
problem.
Okay.
Yeah. Okay.
Okay. Then
So,
time is yours, Dr. Dan.
yeah, thanks.
Yeah.
So, good afternoon, everybody. As I already briefly introduced, my name is Lee Dan. I work at National University of Singapore as a food microbiologist. More specifically, I work heavily on foodborne viruses, and therefore it's a great honor to give a presentation over detection and control of foodborne viruses. Well, due to the time limits, actually, I will be mostly telling over detection of viruses, but in the end, I will have a few words over control of the virus spread as well. I'm actually not quite sure about your background. Therefore, please just stop me during my presentation if you're not clear. Yeah, and we can just have a discussion. Yeah. From the very beginning, very general and brief introduction, what is a virus? A virus is quite different with other microorganisms on this planet, and because it's very small, first of all, it's normally measured in nanometers. It's very small. Is obligatory intracellular parasite, meaning it will never multiply themselves with their host. Right? They have their specific host, but without their host, they will never be able to multiply themselves, meaning on food as well. And so you will never see these viruses be able to multiply themselves on our foods. That's quite different. The second one, since they are very small, they're also very simple in their structure. Instead of having DNA and RNA, and they have only
Excuse me,
one
Dr.
type.
Dan.
Yeah,
Dr.
sorry.
Dan, you speak too fast for our student. Can you be slower? Speak slower. Thank you.
Sorry.
Yeah.
All right.
Okay,
So,
this is not Singapore. This is an Indonesian student. So
yeah, I will
sorry.
try my best. Yeah.
Okay. Thank you very much, Dr. Dan.
It's okay. So our virus will have either DNA or RNA. And so normally other, let's say bacteria or eukaryote cells, they will have both DNA and RNA, but a virus will either have DNA or RNA. So when we are trying to make classification of these viruses, and we would say this is a DNA virus or this is a RNA virus. It only has one nucleic acid. And lastly, the virus is very simple, so it doesn't have a cell wall. Instead, it has a virus capsid. We call it a virus capsid. It's simply a protein shell on outside of these nucleic acids. And so simply just one single molecule of either DNA or RNA wrapped by a virus capsid, so a protein shell, right? That's a virus already. Some of these viruses will also have an extra lipid envelope, but it's not necessarily there. And so it's not a necessary structure. The essential structure for a virus is the nucleic acid, either DNA or RNA, wrapped by a virus capsid. Okay. And then speaking of foodborne virus, as the name suggests, foodborne virus refers to the virus which can be transmitted through food. Not only through food, but can be transmitted through food. And this is how we actually name the virus. We have comment here, or I cannot see the chat, sorry. Is anybody asking question already? No, I cannot see it.
Not yet.
No, I saw the
Not
chat
yet.
was lighting.
Yeah.
But anyhow, back to our foodborne virus, and these viruses cannot multiply in the environment or on foods. Refers to our first slide is because they can never multiply themselves without their host. And therefore you will never be able to see it on the food. Sometimes you see the food is still very fresh, very nice, but it's maybe contaminated with virus already. And so that's quite different from food spoilage. Another important feature for foodborne viruses, all our foodborne viruses we are talking about today don't have this extra lipid envelope. You may say, they don't have an extra protection, so maybe they are more fragile. No, the opposite. Because they don't have this lipid envelope, they are actually all very, very resistant. That's kind of a general rule we observe for all these viruses. For viruses which are naked and without this lipid envelope, they are normally very, very resistant to environmental factors such as heat, low, high pH, drying, and so on and so on. Therefore, they can be infective. They can remain infective on the food for a very long time, and also they can survive our human gastrointestinal system. And so they are very tough because they cannot multiply themselves easily. So they have to be very stable. Otherwise, they die off very easily, right? And once they get a chance to have contact with their hosts, and they will immediately get very, very infectious, they will multiply themselves very fast up to very high numbers. And here I give you examples. So from the human stool or vomit, you can easily have 10 to the 11 or even 12 virus particles per gram. And so this is a unique feature for these viruses in the environment. When it doesn't have its normal host, it just stays stable, like inert. But once it has a chance to multiply themselves, they meet the right host, they start to multiply like hell to very, very high numbers. So we say these viruses are very infectious and they have very low infectious dose, and they can They can already cause infection with very small number, we say 10 to 100 virus particles, and then they can multiply to very, very high numbers in a short period. Most important foodborne viruses, and we normally divide it into two categories. One is on the left side, viruses causing gastroenteritis and either vomiting or diarrhea and so on. And the other group are viruses causing hepatitis, and so problem for the liver, right? And therefore, we have two very representative. And so after this lecture, if you don't remember anything, I hope you still remember these. So the two most representative foodborne viruses are Norovirus and Hepatitis A virus. Yeah. And nowadays, Hepatitis E virus is also more and more getting recognized by the community as a foodborne virus. Why is it important to talk about foodborne virus? And the reason is simple, because it's very, very prevalent. It's causing very high disease burden. And according to the WHO estimation, from the most prevalent foodborne virus, Norovirus, we may have over 600 million diarrhea cases globally every year, right? So this is a huge number, definitely. And in general, norovirus only cause very mild disease. So not a lot of people really die from it, but there are people dying from it. So you do see there's, if you look at the number and think about the individuals, is actually quite sad, and to see quite a lot of people also die from these virus infection as well. Yeah. Now, a little bit general introduction one by one from these important foodborne virus. The first one, of course, no doubt, norovirus. It was discovered from not long time ago, in 1972. At that time, it was named as Norovirus. And norovirus, this name was only made up, let's say made up from around 2003. And so it doesn't have a very long history. Only in very recent years, people found actually is the reason causing a lot of viral diarrhea problem, right? It has many different strains. So it evolves really fast. Every few years, we will be dealing with new strains already. And if you look at the structure, as we just introduced for generally the foodborne virus, it's quite simple, right? It only has a very short single-stranded DNA molecule as a genome, so only 7.5 kb, and it's very variable, and that's why we have so many strains. It only has one virus capsid, like we said, and it doesn't have lipid envelope. And so the structure is clear. It's the same as in general all the foodborne viruses, but it's very stable. It has very low infectious dose to make people sick already from a few virus particles, estimated, of course. And once people get sick from it, and they can shed very high concentration of viruses from their feces or vomits. Incubation period is not that long. Typical symptoms of norovirus infection is nausea, vomiting, diarrhea, and so on. So we call it stomach flu sometimes. In general, you only develop very mild disease afterwards, so you barely even notice it, right? But for some immunocompromised people, they may get hospitalized or even death, and like I just mentioned. And something very worthwhile to point out is quite a lot of people actually still shed the virus after the symptoms. For a long time, sometimes up to a few weeks. This is dangerous, right? We can say, okay, our food handlers, and you have to be careful. You cannot go to work if you are sick, right? But these viruses are very cunning. They are still be shed after the symptoms for a long time. So it's quite kind of difficult to control. Do we have a question here?
Yeah, I have one. So you say that before that the viruses will still eat the gastrointestinal, right? So it still will stay there, although we are already in health condition.
Yes.
In what condition that the virus will come out again, will rise again, and cause a disease?
Normally, it will not rise up again.
Yeah.
Even you will lose your immunity over this virus. That's why till now, Norovirus doesn't have any vaccine. So
Oh, I see.
the virus out there is shedding, but you don't have any symptom. Then somehow it die off, and then your immunity also, you also lose your immunity, and then next time you may also again get infected from the same virus, and also because this virus evolved so fast, so perhaps you will get infected from another strain of these viruses.
So how we can overcome this virus?
Sorry?
How we can manage this virus so we cannot get infected by the virus?
This is the purpose of my lecture today.
Oh, I
you
see
have
that.
to,
Okay,
you
then.
have to keep on listening then.
Okay, please continue then.
Yeah, okay. There are still some other foodborne viruses, of course, causing gastroenteritis, but it's less important. It's either less foodborne or less severe. So we will here we will not talk about it one by one. Hepatitis A virus, I believe most of you are more familiar with it. Simply because it causes more severe disease and it has a longer history. It has similar structure with norovirus. Good news is it doesn't evolve that fast. So for hepatitis A virus, we don't have that many strains.
Yeah,
Okay.
good news. Yep.
Yes. Therefore, we have effective vaccine for hepatitis A virus. No much to say. Hepatitis virus still follows the same structure, as I said, a single positive stranded RNA wrapped with this virus capsid and no lipid envelope. Okay. Infectious dose is also low and it can also be shed with very high level. Incubation period is much longer, up to a few weeks. And the symptoms, of course, are more severe, mainly infecting the liver. Also, something interesting to share is for hepatitis A virus, normally we believe we see more severe symptoms from adults than children. So for children, let's say younger than 6 years old, they can get the infection, but the chance to develop asymptomatic infection is higher. Okay, so that's overall the trends we believe. And in nowadays, we found this trend is changing somehow due to the improvement of hygiene situation, we believe, because you also actually see in developing countries, you have actually less hepatitis A virus infection problem than developed countries with better hygiene problem. Why? Is because in developing countries, you have more children actually already get exposed to these virus when they were young. And therefore, they will develop asymptomatic infection without knowing even, and they will develop lifelong immunity. And therefore, when they are adults and they grow up, they still have this virus around them, but they are simply resistant to it. And in comparison, in developed countries, they're very careful about their children. Overall hygiene situation is better. So their children normally are not immune from this virus. So when they grow up, and if they by accident consume food, for example, with hepatitis A virus, they will develop a more severe illness. And nowadays, of course, and overall speaking globally, the hygiene situation is improving. So we say this virus are also getting smarter somehow. is trying to infect children as well. So we see virology is like moving, is dynamic all the time. It's also, it's like the COVID-19 is trying to fight against the human being all the time. Hepatitis E virus, as I mentioned, also in recent years recognized as foodborne virus. Its clinical features are quite similar with hepatitis A, except that for hepatitis E, you see some geographical distribution pattern and from this one. So for certain strains, it's more prevalent in certain areas and is a zoonotic pathogen. And so for both noro and hepatitis A, we know if we are talking about the human strains, they only infect humans so far. Well, they are still evolving. We don't know. So far, all of these human strains we found for norovirus and hepatitis A virus only infect human beings. But for hepatitis E virus, it's very different. It's clearly a zoonotic one. So we have the same strain, which can infect both human beings and animals. And we think all these animals, pigs, are the most important ones. And therefore, pork meats are kind of the most closely related food type with hepatitis E virus. That's very important feature being different with the others. Okay. Transmission of question? No. Okay. Transmission of foodborne virus. As I said, foodborne viruses were termed as viruses which can be transmitted via food. So mostly we see food are involved in the transmission, but I'm not saying that's the only transmission routes. Actually, transmission routes of foodborne virus can be very complicated. The most effective one is still person to person. Here can also be transmitted via water or environment surface, and so on. Of course, food is also involved. We can roughly divide the food-related transmission into two parts. The first one, we call it pre-harvest contamination, meaning this contamination happens during the pre-harvest stage of the foods. For these contamination is normally due to water, due to contaminated water. And the most important food products involved are, for one thing, shellfish, such as oysters. Why? Because these are the filter feeders, and how do they get food from the water? Is they try to filter large amount of the water in order to get the small particles to eat, right? And unfortunately, these viruses can also be accumulated in their gut and get protected and stay there, very stable. They don't multiply, by the way, as I just said. They don't multiply, but they can be accumulated and protected in the gut. But it's just firmly attached, and it's quite difficult to get rid of them. Even perhaps it's inside of the tissue. And lastly, very important reason for these two types of foods and being associated with the outbreak is because people like to eat them raw, right? You don't want to cook your oyster that much. You also don't want to cook your lettuce or berries that much. So this is also an important reason. Transmission via food. The second one, we call it harvest or post-harvest contamination. For this one, is mainly due to the food pickers or food handlers, right? Because they are infected, and some of them may have poor personal hygiene, and some of them, for any reason, they come to work when they are ill. Either they have to, because they have to make money, or they are asymptomatic shedders. So they don't even know, and nobody knows, like I just mentioned. So if they don't really follow strictly the hygiene rules, so they, after visiting the toilet, and they may have large amount of viruses on their hands, so it's easily causing foodborne outbreak as well. Okay, now finally we come to our main topic today, detection of these viruses. Being quite different with other foodborne pathogens or bacteria, in general, all our foodborne viruses remain very, very difficult to be cultured in the lab. Doesn't matter what you are using. You are using cell culture, stem cells, even 3D cell culture, animal models, all very, very difficult to cultivate them unless you use human volunteers. But that's extremely difficult, right?
Yeah.
And therefore, for food virologists, it's almost impossible to get to culture them and get to know their real infectivity. So that's a big problem. On the other hand, we do need to detect them. We don't need to look whether this food is safe or not, right? And therefore, molecular matters is almost the only possibility for the moment to detect them. And please note, it's for food virologists, right?
Yeah.
If you are looking into this clinical virologist and they say, we still have other matter, we have immunological matter, by the way, it has really low sensitivity, yeah, and so on and so on. So I would say for food application, molecular detection methods is currently the only possibility to detect them. And since we cannot culture them, since we will require very high sensitivity, as I will explain you later. Around 15, 20 years ago, it's when I started to work on these viruses. It was a time most of the labs globally are transferring the use of traditional and conventional PCR, like running this gel, looking into the bands, into the use of real-time PCR. Therefore, real-time PCR is not a new technique anymore. That's many, many years. Right. At that time, people were very happy about this method. So real-time PCR, let's say 10, 20 years ago, quite trendy. People are very happy, excited about it and say it's super sensitive application for virus detection also, and they start to make standard methods and so on. So as a first step of introducing you the detection methods, I will briefly introduce you the basic procedure of virus detection from foods based on PCR, based on real-time PCR. The first procedure is sample preparation, the same as all of our food detection, right? And we are talking about molecular detection, we are talking about all kinds of detection methods. Actually, there is still a very long way to go before you can really reach to the step to perform your detection. You have to deal with food first. The sample you receive are always foods instead of looking into these biological samples, right? As I can tell you, it's very, very challenging to concentrate and purify these tiny, tiny virus particles in very small amounts from so big amount of food with so complicated components, right? We have lipids, we have carbohydrates, we have protein, we have, so complicated, right? From the foods. And then in the end, we may only have a few virus particles on top of it, which cannot multiply without the host. So it's really, really challenging parts that if you want to collect it in your tiny tube. I will not go into the technical details, but overall we have a few categories of strategies. Let's say sometimes most commonly used is elution concentration. So we try to wash off the virus particles from the food, and then we try to concentrate it in the washed off elution. We can directly try to extract all of these RNA. So we extract RNA even from the food. That's kind of stupid methods, right? But that's really for some very complicated food matrix that we don't have other solution, and you cannot wash it off, right? We also have Pronase K plus heat treatment as a very, very simple methods, but it can be only applied to shellfish, very, very specified, because shellfish, they accumulate viruses, like I just mentioned, in their guts. So you already have highly accumulated viruses in their guts. So it's like the shellfish are helping you to do the detection. And therefore, for these shellfish, it's kind of comparatively easier than other food matrix. But anyhow, the message I want to say is, bear in mind, this is the bottleneck for detection. This is the most difficult part in our most challenging part in our detection procedures. Secondly, after you try to catch the virus particles from the foods, your next task is to purify, or let's say extract the RNA. That you remember, this is the structure of a virus particle, capsids, and inside is an RNA molecule. So we still have to extract the RNA, right? For this part, it's generally easier. And nowadays we have a lot of commercial kits or even automatic systems. That's quite easy to do. And lastly, finally, we come to our molecular detection. There are kind of many different types of molecular detection methods developed in recent years, but I can tell you RT-PCR representing for reverse transcription polymerase chain reaction, RT-PCR is still the golden standard. And no other alternative methods can take the place of this method still. Why we do RT-PCR instead of PCR? That's because the genome of our foodborne virus are RNA. And in order to carry out a PCR, you will need a DNA as a starting template. And so we'll have to have an extra step, reverse transcription, and to prepare our DNA copy from the RNA. And nowadays, quite often, we have already reagents to combine these two and to perform experiment altogether. And just one master mix, and then you can first perform your RT, your reverse transcription, followed by the PCR. The reason I said why people were so excited that we can use real-time PCR instead of conventional PCR is because with the use of this probe, let's say here we are introducing basically the TechMon probe-based real-time PCR. It's because we can follow up the generation of double-stranded DNA. And here, I don't know whether you all have this background knowledge over PCR, so I'll briefly introduce and the procedure of a TechMon probe, real-time PCR. So here we have a double-stranded DNA or as our template, right? We will heat it up, normally to 95 degrees or something. Then this double-stranded DNA is denatured into two single-stranded DNA here, okay? At this time, we change our temperature. This temperature is adapted with the primer we designed and synthesized. So this primer is actually designed based on the sequence of our targets, right? And the sequence of the primer will define the annealing temperature we use. So now we lower down the temperature to the annealing temperature defined by the primer, so that the primer and the probe will anneal, attach to this single-stranded DNA, okay? And a normal conventional PCR will only have these two primers, forward and reverse primer, but a real-time PCR with TechMon probe, we have an extra probe. Why this one is special? It's actually the same with the primer, just a very small segment of single-stranded DNA, but it's also linked with here a fluorescence reporter and a quencher here. And please note, if these two small groups, fluorescence reporter and quencher, are linked together with this probe, nothing will happen. There will be no fluorescence because of the quencher is here and they're all linked together. So this one is muted. The fluorescence is muted. So basically you don't see anything, okay? They are there. They are silently binding to the single-stranded DNA. And next, if you change the temperature again to around 60 degrees with the help of polymerase and these enzyme, then elongation starts. It's like two trains and starting from these two sides forming into two double-stranded DNA, which is the normal procedure of conventional PCR. But remember, here we still have something special. We have this probe sitting in the middle, right? Sitting in the middle. So when this elongation go on, and reach the position of the probe, it will be hydrolyzed. Okay, and once this probe is hydrolyzed due to the formation of double-stranded DNA, this fluorescence group will be set free to the system. And here, without the limitation of quencher, they're all set free, and now they are set apart from each other. This reporter is now giving signal. It's giving fluorescence signal. And therefore, we simply need to collect the generation of this fluorescence signal. Then we know, okay, each time if this fluorescence signal is increased, our formation of double-stranded DNA is also increased. Okay, and here on the right is a typical raw data you collect. And see, here is the fluorescence. Y-axis is the fluorescence signal that you go on, and PCR cycles go on the time. Each time after one cycle, you collect the fluorescence, and you typically will get a curve like this. Okay. And then at this time, you simply need to set up this threshold. Normally, the machine will set it up for yourself automatically from this exponential growth phase. You set up this threshold and you say, okay, now you start to compare between samples. You see the green sample, I would say this green sample, which has a high virus concentration, it will need less PCR cycles to reach this level. Of fluorescence. And in comparison, if you have less virus concentration in your sample, you will need more time. You will need more PCR cycles to reach the same level of threshold. Therefore, simply by comparing the time, by comparing the PCR cycles, how many PCR cycles do you need? Does this sample specifically need to reach this threshold? And by noting down the CT value and cycle thresholds, how many cycles do I need to reach this thresholds? We will be able to compare the concentration of the starting material. That's originally I have more virus in it or less virus in my sample. So this is the basic principle on how do we quantify, how do we follow up the generation of the signal. And if you want to do absolute quantification, you simply need to get your standard material with known quantities, generate a standard curve. Actually, that's the same as everything you're doing to quantify also your chemicals. And lastly, our story is not done, of course, and you say, okay, now you're done from the sample preparation, RNA, and then we do the real-time PCR. We get our signal, yay. No, it's not done yet. The same as all our other microbial analysis, you still have to set up a lot of controls to make sure your positive is really positive and your negative is really negative. It's not. It's only because of your sample is positive or negative. It's not because you made some problems during this procedure. How do we do that? We can set up many different types of controls, and we can give them different names. And here I summarize 4 categories just to give you an idea. For example, we can set up the first category of controls. We name it process control. What does it mean? It's also a virus. It's not a virus we would like to detect. It's normally an artificial virus. We know for sure it's not present in our food sample. And also, we know the quantity of the virus for this process control. So we add a known quantity of this artificial virus as a process control to our foods. And then we perform all the procedures I just mentioned, all the way in the end into real-time PCR. So when you're doing real-time PCR, not only you detect your target, but you also try to detect your process control virus. And remember, we actually know how many viruses we added in. In the end, you will generate a result. For example, you add 100 copies of your process control virus in it. In the end, you found you got 2 copies only. Then you know your recovery is 2%. So you lose 98% of your viruses during this whole procedure. You don't know where, but you lose 98%. Okay. The second type, we call it RT-PCR internal control, is similar with the process control, but is added from a different procedure. So you add a known quantity of external RNA into this RNA extraction, and you also perform an RT-PCR out of it. And again, suppose you added 100 copies here, and then you detected, let's say, 50 copy, then you know, okay, I can only detect half of my genuine virus RNAs in it, ideally, right? Thirdly, we call it PCR positive control. It's simply to make sure your PCR is running smoothly. Otherwise, if you forget to put any components, and then everything is negative, right? So you have to put some positive control and to make sure your PCR is running smoothly. And similarly, PCR negative control, you just add some water as your template to run a PCR, and somehow you may find, you detect a signal from water. That's wrong, right? That means you have contamination. So for all of these scenarios, for all of these, for both, let's say, positive and negative control, if you have problem, then you know, there's something wrong with your experiment. You cannot trust your results over your samples anymore. Yeah.
Okay, sorry to interrupt, Gudnan. This is one question from Pak Cahyo, from Mr. Cahyo. The question is how do we address the issue on false negative result during detection, which caused by the low concentration of RNA virus? The virus is there, but very low concentration, and then the RT-PCR failed to give result.
We have some impatient audience. This is indeed, again, very good question. This is a problem. Yeah, this is my point. Real-time PCR is supposed to be a very, very sensitive method. This is also the reason why I keep on saying sample preparation is a very challenging step, because indeed, if you have very low amounts of virus contamination, you simply cannot detect it. The sensitivity of these methods, if you keep on listening, you will know it's actually not high.
Okay.
Can I go on?
Yeah.
Okay, so I also followed by that question. You may be curious, like why I give these examples and process control. I give actually a very optimal example. I said you can recover 2% of the viruses. Actually, the reality is ISO definition is as long as you reach 1% recovery, you already pass. So the standard there is the recovery has to be higher than 1%. But the reality is quite often you don't even have 1% recovery. And this is also, again, why I say the food preparation is quite challenging. So in the end, if you can recover 1% of the virus contaminated on the foods, it's already a great success. All right. And then, of course, here this PCR problem is less. And so I give an example of 50%. The criteria is actually 25%. So if you add some RNA in it together with the food matrix extraction, then you do your PCR, you will normally have higher than 25% recovery from the RNA you added. All right. So my story is definitely not there yet. It's still at this very early stage, let's say 10 years ago. At that time, and these methods were already developed, and standard methods were already being discussed. At that time, we didn't actually realize the sensitivity is not high. At that time, we were really happy about these methods. Therefore, at that time, people were talking about the other problem of a PCR. Because as you may realize already, if you perform a PCR, what exactly are you detecting are a very small segment of the RNA, right? Around 100 BP. It's not the virus itself. It's surely not the alive virus. Meaning in the end, if everything is fine, You have a positive signal, all your controls are fine, but what are you detecting? You can only say originally, I have this virus RNA, and perhaps not even intact molecule, perhaps it is even a very broken segment over there, and by any chance it's just my amplicon, it's just my target, then I pick it up, I get a positive signal, right? And this is a problem. At that time, people were talking about, we say, okay, real-time PCR is indeed sensitive, but maybe it's too sensitive. And maybe it's only detecting these broken RNA, and we cannot really trust these results correlate directly with public health. So what do we do at that time? And then therefore we are trying, from my PhD study, actually at that time, we were trying to lower the contamination, not contamination, lower the false positive result, possibly from these broken, or let's say dead, inactive virus particles. A lot of different, here I give you again more concrete examples. And suppose here you have intact virus particle. Of course, that's very high chances infectious. But imagine if you have a heat-treated virus, and this capsid, this protein is broken, but your PCR target is still there, right? And even if you use UV to treat it and to damage the RNA, quite possibly it's damaging other parts, and quite possibly your target is still there. So it's quite possible if you just do a real-time PCR, even if this real-time PCR is done perfectly, but still it may, theoretically, it may still give you a lot of false positive results from these examples. And so we were trying to develop a lot of methods because we cannot culture it anyhow. So we can only try to develop some modification of these molecular methods to give some prediction of these virus viability. For example, I only give you one example, is these RNase treatments. So we imagine if this virus is intact, is complete, so these RNAs very well protected. If we add RNase, it will not have any chance to digest the RNA here. And in comparison, if you indeed have a broken virus particle, this capsid is broken, then this enzyme will have its chance to enter the capsid and to digest the RNA. And therefore, by this way, you will be able to decrease somehow some of these false positive results, right? And due to these broken inactive viruses. And there are still quite a few examples, but the problem is we develop these methods and we try to apply it to real foods, right? We develop these methods, then of course we use these artificial samples, but then afterwards we say we would like to apply to real foods collected from the markets. Then we actually found that problem. Then we found, the real-time PCR detection is not that high. And so ideally we would like to use real-time PCR to select some foods with high contamination level. Then we apply our pre-treatment methods such as these RNase methods and to lower the false positive results. And that's the ideal idea from that moment. But then we found we already have problem from the first step. And we want to find the baseline real-time PCR detection. And then we already found very, very low level. For example, here, we together with Nestle, we carried out a very big surveillance over 2,000 samples. And we only found 7 samples positive out of it. And all of the positive samples of this with very high CT value. Very low level. So you see, we simply don't have any space to apply our pre-treatment method to further lower the false positive results anymore, because the baseline is already low, too low. There's no space for us to make it even lower. It's actually at that time, we start to realize, actually maybe our real-time PCR is not that perfect. It's not that ideal as we imagined. Why? Because these outbreaks keeping on popping up and reminding us, here you don't find a lot of positive. It's not because your viruses and your foods are indeed safe, because these outbreaks keep on popping up still, and there's still a problem with this contamination. It's simply because your methods is not sensitive enough. So it's actually only at that time, if you remember from my first few slides, the starting time of making up these standard methods is 2004. And the first edition of these standard methods was only published in 2017, out of 13 years of discussion and all these back and forth things like I just mentioned. Sounds ridiculous, but it is like that. I can tell you, even if this ISO method is finally published, this method still suffers from many, many uncertainties and limitations. And the most important limitation, as we already discussed a bit just now, is the sensitivity. As we mentioned, ideally, a real-time PCR reaction is very sensitive, is able to pick up one single copy, right? But it's from where? It's from the PCR tube. A PCR tube is quite small. In order to do your PCR, you can only load a few microliter of your DNA. Or let's say if you're doing one-step RT-PCR, it's your RNA. You are only allowed to load a few microliter of your RNA. And above, as we just explained, we have our virus extraction, we have our RNA purification, we somehow still dilute our RNA in order to get rid of the food interference, and we run our RT-qPCR. Each step, we can only take a few proportion from our last, and the results from our last step. And therefore, in the end, you're actually diluting a lot by doing so many steps. In the end, suppose your detection limit of a PCR reaction is indeed 1 copy. But if you want to calculate back to how many PCR copies in your 20-gram food samples, it's quite easily you have rather high detection limits. It's a lot higher than the infectious dose. Remember, I said 10 to 100 virus particles can make people sick. And now our theoretical limit of detection is actually above this infectious dose per 20-gram food. That's not a big portion, by the way, right? So you already have an idea whether PCR is sensitive or not when we are applying for this application. I'm not saying this method is bad, but I'm only talking about this application. The story is not done yet. I have some even worse information is just now, this limit of detection I mentioned is theoretical. It's calculated by the method itself. So each time I take a proportion to do this next one. So for sure, I end up with this much. So, but this calculation is based on what? It's based on each step you recover everything from the food. I did already mentioned the recovery is actually very low. This is from ISO. The criteria is only 1%. So in reality, in practical, the recovery is even lower, meaning the practical limit of detection is even lower. Some labs perform experiment to see, okay, what's our real limit of detection, meaning we spike certain viruses and we try to recover these viruses from these specific food types. In the end, we found this practical limit of detection can be up to thousands of genomic copies per food sample. And again, remember our low infectious dose from these viruses. So this is only one aspect from this limitation of these methods is the sensitivity. We still have uncertainties and so on, and due to the time limit, I will not talk too much about it, but just to show you the limitation from the sensitivity part. Do we need a break, or I can just go on?
Up to you, Dr. Dan. I'm still waiting for any question from the participant here. Now the participant already increased about 155 people.
Okay. Yep. I didn't realize we have so many.
Yeah.
Okay.
So if you
any
want to
question, because this is kind of the end of the real-time PCR story.
Yeah. So anyone? Not yet.
Okay, then I will go on. I don't have too much anymore. So you may ask me now, okay, sounds very sad, sounds very limited. And what's your solution for this detection? Well, no much solution. So that's kind of the history I have been telling you. We had conventional PCR, we had real-time PCR afterwards, and then we were talking about this false positive a lot. You see quite a lot of papers publishing during the years around 2010. And then back to very recent years, from let's say around 2015, we are back to real-time PCR and talking about the low sensitivity and so on, very back to the basics. But we see it's actually still a problem. Again, it's actually because these viruses cannot be cultured. And for bacteria, it's actually the same. If you don't make enrichments, it's actually the same. It's quite difficult to find out foodborne pathogens from food samples. But many of the good news is for many of the foodborne pathogen like Salmonella, like E. coli, we can do enrichments and we can let them grow to very high levels. Then it's easier, much easier to find them, right? Nowadays, well, people are talking about digital PCR sometimes, and maybe it's a better choice. There is still no consensus yet. The reason why people would like to use it is, for one thing, it is an absolute quantification. So something I didn't mention about the real-time PCR in using in quantification is you simply cannot really trust the quantification, especially at the low ends. So with the use of standard material, generate a standard curve, the quantification results easily shifts, and with, well, depends which system you use, the standard material you use, even the reagent you use, it really shifts a lot. And when it comes to the low ends, it's crazy. And therefore, digital PCR perhaps supplies a better solution, because it's an absolute quantification. Did I prepare? Yeah. Why? Because in a digital PCR, instead of using standard material, to generate a standard curve and so on, we are actually separating our reaction into many small units, either on the chamber, like this Thermo Fisher system, or so they have these chips. They have many small chambers here, or this BioRat system that has many small droplets there. So ideally, theoretically, in each of these small units, you will either have 0 or 1 of your molecule, of your targets. And then afterwards, you will run your PCR, and you will count how many positive units you have. Actually, it's not you count, it's the machine counts, thanks to technology involvement, of course. And our machine can count so many small units, and by giving fluorescent signal. So if we have one single positive unit, that means we have originally had one copy as our starting material. So this is achieving absolute quantification, let's say. Okay, so this is already improvement in comparison with real-time PCR. We get rid of the bridge, and the bridge is the standard curve. We get rid of it, and here we can look into it directly. And secondly, we believe digital PCR somehow are more tolerant to inhibitors is because, again, they are separating the whole system into many small chambers, and somehow, yeah, overall speaking, statistically speaking, the interference in each small units and could be smaller, and because your interference can be distributed in somewhere else. But I have to be honest with you, it's not really conclusive. And digital PCR for the moment is much more expensive. Real-time PCR is already expensive, by the way, and digital PCR is even more, and is more time-consuming. And therefore, we believe, well, you can still do the first-line screening with real-time PCR. And if you found certain samples really of interest, positive and of interest, that you want to know the quantity, you can do a further step and to apply the digital PCR and to do a quantification. That's kind of the status for the moment for digital PCR. But of course, this is really not the final solution because it still suffers from many problems. And although ideally I tell you, and it's either 0 or 1, but in the end, you actually see also a lot of positive results. Well, difficult to tell. It's in the gray zone, and it's difficult to tell. Okay, this fluorescent signal should be regarded as negative or positive. Still quite a lot of technical problem to solve from the digital PCR, but this is kind of a trendy technology we are talking. Hopefully to solve some of the problems. So in summary, that we talked about the overall current development of foodborne virus detection from food samples, and the history has been there for over 20 years. Finally, we have this ISO method to be used. It's surely an enormous step. As I said, a lot of people have been working on it for over 10 years, a lot of bitter discussion. Each time we go to a conference, there's always a discussion over that. Yeah. But still, generally speaking, the virus detection is still rather expensive. You will need quite skilled personnel. Overall speaking, if I need to train a technician or a student that can trust the result he or she generated, I will need around like a month. So for short-term students, for example, I will never let them do this because the result is not really trustable. Yes, please.
That's all right. Just finish your summary detection.
Okay. Then in the end, of course, as I just pointed out, it still suffers from multiple uncertainties. Yeah, so that's kind of the current situation for detection. You may ask me why, like you are really disappointed, like, oh, then why do you do that? It's so expensive, right? So expensive, and the performance is still not good enough. The answer is we don't have other solution, and the real point of applying our screening program or surveillance program is for protection. So this is actually a very important concept from our food safety domain. Is always much more important to prevent contamination from occurring than trying to detect it, trying to solve it after the contamination. So the point of performing the surveillance, doing this detection, is not for example, batch release purpose. So we already produce a batch of foods and we do a detection, and then we found they are positive and we try to reject it. No, that's not a good way. That's really old school. It's not effective. The good way is we have a food chain and we establish either ad hoc or a routine detection program. And once we have a problem, although we realize there are still some problems over our methods, but we are already trying our best, right? But imagine if you once, from either these ad hoc or routine detection, you found a problem, you found a virus detection, you found a virus positive signal, then it's the alarm for you to look back, to check your whole food chain, right? To see, okay, where could be the problem? Where could be the contamination source? It's much better, it's much effective than just doing nothing and wait till you have final products and then to let other people find it out and then you simply, you know, get ruined, right? So that's the real point for detection. Um,
sorry. We have some question here.
Yeah, please.
Okay. So,
I still cannot see the chats.
yeah.
Where did you get?
To everyone, I think, in the meeting. Yeah. Similar. I didn't see either.
No.
Yeah. Actually, before we started this, your
Next
lecture.
Question. Next question,
Yeah.
Parsa.
Yes.
I written it in the chat. Could I mention what our question?
Sure.
Yeah. To Prof. Irma. Please
Thank you.
go ahead.
It's very interesting explanation and lecture today. I am very happy to being in this lecture. And I would like to know what kind of updated research that is conducted now by your university, maybe related to the first one, foodborne viruses, and the second, COVID-19 virus.
Sorry,
And
sorry, I didn't really get you your question.
Okay, I would like to know what kind of updated research of your university.
Accreditation.
Updated, yeah.
No,
your research today about foodborne virus and COVID-19 virus.
COVID-19. Oh, wow.
Okay.
Yeah, a lot of people start to ask about it.
Yeah, I would like to know how the university can give contribution in the COVID-19 prevention.
First of all, we are talking about foodborne viruses today.
Yeah.
COVID-19 is definitely not foodborne virus. So it's very, very different, I have to say.
Yeah.
So it's surely not my job to look into COVID-19, this virus, and because it's really, really different. But secondly, since you bring it up, nowadays there are still quite some discussions. Actually, I just published a review paper to talk about this issue. A lot of people start to ask whether COVID-19 could be transmitted via food. Right, because you really read from the news, like somehow COVID-19 can be transmitted from food or food packaging materials. Our major opinion is COVID-19 is not, it's for sure should not be regarded as foodborne virus. Why? Because as I mentioned, foodborne viruses are viruses mainly transmitted via foods. And also we believe there's an important condition that these virus cause infection should be able to cause infection due to direct oral injection, if you know what I mean. So COVID-19 doesn't really follow these oral fecal routes. It's not like norovirus, for example, the patients shed it from their feces or vomit, and then people out of all these transmission procedures, they eat it. It's via the digestive tracts, right? So here we still believe it's somehow, some careless contacts. It's not really foodborne, so it cannot be attributed to foodborne. On the other hand, somehow it can be regarded as occupational disease, because if you are working with, for example, these farmers, if they irrigate their vegetable, and now we already know these sewage or surface water has sometimes quite a lot of COVID-19 virus, they may have more contacts, exposure to these viruses. But again, it's regarded as occupational disease. So since you bring it up, I would like to clarify.
Yeah.
This is not really in the zone of foodborne virus.
Yeah. So, Dr. Dan, I would like to ask, what is your recent work? I mean, your university work in foodborne virus.
I actually work on everything over foodborne virus
Mhm.
detection, inactivation. That I do tell a little bit afterwards. And also I have interest in the consumer ends of these virus. Yeah. As I just mentioned, out of so many years of work on the virus, I still believe the most important thing is human being is actually to control human behavior. And once I was talking to a very known professor in this domain, and he said, you know, as long as you can control human fecal material, then there's no problem for human norovirus anymore. But unfortunately, you cannot. It sounds crazy, but it is a worldwide problem because of these basic human hygiene practice. Yeah. Is there any more question, or I can finish my?
I have questions, Doctor.
Yeah, please.
If you don't mind.
Yeah, please.
Okay. First thing is, if you compare, let's say, plant and animal-based foods, which one is actually more prone to virus infections, based on your knowledge, let's say? Are there any trend where the animal, I mean, meat, milk, or the plant-based foods are more more sensitive toward the virus, or?
I'm not sure if I get your question or not, but the only very known zoonotic virus is Hepatitis E, as I mentioned.
Mm-hmm.
Yeah, so for norovirus, for example, as I mentioned, it has many different strains. It has four different genome groups, and you do have norovirus for many different animals. And we have mouse, we have dog, we have pig. Many different animals can be infected by norovirus, but it's not zoonotic, meaning human strains only infect human. So far, we found. They still don't cross the boundary. But of course, there's always a chance in the future. There's only this Hepatitis E virus is very zoonotic. Yeah.
Okay.
Yeah. So mostly from domestic pigs.
Okay, Dr. Dan.
Yeah.
Sorry. I think we have some question from our student here, from participant. Can I call Desi Anggraini Sutanto?
How do you see it? I don't.
I have a chat in here.
I don't.
No, you don't?
There is a
No,
chat
I don't.
icon below your screen, Doctor.
Below your screen.
Yeah, I know how to use it. I have the chat, but I click it. It doesn't give me any.
Oh yeah.
It doesn't give me the window. Strange.
Okay, anyone from Desi Anggreni Sutanto or Adib Zainuri or from Yolandani? Anyone? Or I see already someone raised the hand.
Hello, it's my first start.
Oh, is it Adib Zainuri?
Yeah.
Okay. Please go ahead.
Yeah.
Go ahead.
Hello, Dr. Dan. I'm Adib Zainuri from IPB University. I want to ask about the PCR cost. Is it maybe there are any other option to detect virus or bacteria from food rather than using PCR? Because we know that PCR cost is very expensive. Yeah, thank you.
As I mentioned, there are a few alternatives like LAMP. LAMP was developed a few years ago, well, not a few years ago, also over 10 years ago. Alternative methods. But I'm afraid, you see, even PCR, the better methods already suffers from many limitations. Yeah, so.
Okay, thank you for for your answer, Dr. Dan.
Yeah.
Okay, thank you, Adib. Next, Desi Anggraini or Yolandani?
Okay, may I ask?
Who is it?
My name is Yolandani.
Okay, go ahead.
Okay, Prof. Dan, my name is Yolandani from Jiangsu University in China.
That's also one of the most challenging thing you have to solve from this problem. But that's again not only for foodborne viruses, that's actually for all of our foodborne outbreaks. Source tracking is normally quite challenging. Since you have underreporting, you have different it's just imagine if you have an outbreak, how do you actually identify this outbreak? And once this outbreak is really or water or surface, let's say, is possible. But meanwhile, people are indeed doing a lot of efforts each time. If there is an outbreak, the government will for sure have the action, in a very systematic way to do the source tracking. And due to the source tracking, of course, you will have different types of samples. You have not only food samples, but you also take samples from the personnel, like you take from their hands, from their fecal samples, from the surface. So you have swab samples, you have
Yeah,
water
kenapa,
samples.
Mas?
Yeah. Do I need to stop? Or
Oh,
hello?
hello.
Proceed, Dr. Dan.
Yeah.
Go on,
Please proceed, Doctor. I think something technical issue with the moderator.
Oh, okay, sorry.
Okay, Prof. Dan.
Yeah.
So, when you do an experiment, so what do you usually do? Like, do you also do a pre-experiment or just do the PCR directly in for your experience? I mean,
what do you mean by pre-experiment?
Like to the screen, like the source tracking. Have you ever like do that source tracking?
Oh, that's not my job. No. I don't have that chance to do that. I would like to, but there are always a lot of political issues there. I will not have the chance to get contact with these outbreak scenarios. So once outbreak is reported, of course, it's the government who are there. It's not our researchers.
Okay.
Okay, I got it.
I will be happy to have some samples like that.
Yeah,
Okay.
it's really challenging.
Yeah, you have to really build up good collaboration with people from the government, from the hospital, and so on.
Okay, thank you for the answer, Prof.
Yeah,
Okay, Dr. Dan.
Yeah.
Sorry, but please finish your presentation.
Yeah, indeed.
Yeah.
I also hope to.
Okay, thank you.
Yeah, sorry about my typo here, but here I was actually thinking to write control. As I already mentioned, it's not really possible to finish all my story over foodborne virus, and so I decided to mostly focus on the detection part. And then over this inactivation, we also put quite a lot of efforts in the virus. But in general, if you talk about virus inactivation, actually it's quite simple because all our viruses are not heat stable. If you just warm up our foods at till 70 degrees for a few minutes, let's say, then they're all dead. So no issue. But the problem is for many of the foods, as I just mentioned, raw oysters, fresh produce, people don't like to heat it, right? Therefore, when foods are involved and thinking about the texture, the taste, the color, freshness, all these organoleptic or nutritional requirements, don't always want to warm it. Also, if I talk 60 degree and you think, oh, that's almost nothing, right? We all it with high temperature and cook our foods very well with high temperature. But if I did you actually maybe not. We once did it once in a lab. We took a piece of meat, it's like beak. It was for chicken, I remember. And then we bake it. Well, in Asia, we don't often, but in Europe, they do it very often. They just bake a very big piece of meat with a pan. And this really looks like well done, and both yellow, both sides yellow and so on. And measure the core temperature is still somehow not really 70 degree yet. So the temperature thing is actually also tricky. By the way, therefore, in order to fight against virus contamination, people actually develop many different mild and non-thermal treatments. But meanwhile, you have to make sure you don't bring toxic residues. You have to be natural, you have to be sustainable, and so on. It cannot be too expensive. I myself have been trying many different treatments, starting from high pressure processing over oyster. Then we look into fresh produce like hydrogen peroxides, UV treatments, these natural components like grapeseed extract, and so on and so on. Overall, I would say today I will not give these exact examples anymore, but overall, I can tell you my experience and opinion are you are using mild treatments, then you don't actually expect a lot of reductions. That's overall the message. So none of these mild treatments, I can tell you, is really as effective as heating. So somehow it always gives some reduction, but none of this treatment I can recommend to you today, like, hey, if you suffer from this problem, use this treatment and you will get rid of it. No, it's always quite limited as well. We try to use sometimes use some combination, so we can have hurdle reduction. And also another problem in our inactivation study is, again, due to these virus cannot be cultured. So if you would like to carry out a research evaluating whether your viruses are killed or not by these technologies, you will at least have to build up a good methods, right? And to tell, okay, now the virus is alive, now the virus are, let's say, 2-3 log inactivated. But our foodborne viruses cannot be cultured. Then how do we do a good evaluation? You can say, oh, you just set PCR methods. Yes, PCR methods is surely not sensitive enough to do this evaluation because especially you treat your food with these mild treatments, the RNAs are always there. So you will never see a reduction from these treatments. Therefore, you see the problem on one hand, the treatment itself is not that effective, let's say. On the other hand, the evaluation system is not that mature or easy to use. There's always a lot of space for discussion. So that's kind of, the overall progress for these virus inactivation as well. Still a lot to do, but we are just waiting for breakthroughs. For the most important one, Norovirus, it's the enteroid culture system was successful 2 years ago, but it's still very expensive, very difficult. Each time you have to have organoids isolated from patients, and this system will need very complicated culture system. You will need to culture many different other cell line to nutrient this enteroids, and the efficiency of multiplying human norovirus is also quite low. And you can actually not multiply all of them. Only a few strains are successful for the moment. So still a long way to go before we can easily evaluate it or make use of it in our foods related research. So over till now, my take home message for you guys is yeah, don't be too sad. Don't be too pessimistic over this virus research. It is like this is it's quite simple, it's quite small, but it's really difficult to deal with, right? All the stories I tell you, and if you ask me now, then what's the solution, right? As a food safety researcher, what's your suggestion to mitigate it? My answer, as I just mentioned, is all prevention. That's definitely more important than trying to inactivate it when it's already contaminated on your foods. And as the governor, you can say, okay, do detection, and then I will decide whether this food safe or not. Can tell you this is definitely not an effective tool. And just one more word, this batch release strategy is also not effective for other pathogens. Even if your methods is better than this methods I'm currently introducing, you still suffer from the limitation of sampling. You can look into the statistical aspects of sampling. You say, okay, large chance if you do this batch release that you actually let a lot of contamination go and let our people exposed from the risks. So in order to enhance the food safety overall, not only for foodborne virus, but for everything, for all our foodborne pathogens, the most important strategy is always prevention. For example, again, together with Nestle, and we develop this small booklets and to guide the farmers on how to mitigate, to minimize the risk of microbial contamination. It's very much in detailed. It's very easy to understand, starting from like growing fields selection, animal regulation, domestic animal, wild animal regulation, manure-based soil amendments, agriculture water, which is essential, right, to always bring contamination. Also for food handlers, our working harvesting practice all the way to storage, transportation, and then it's the most effective one is always to look into this practical thing, this detail. Sounds like common sense, but if you really look into it, it's not really well done, right? Out of the real life problems, right? And also for food handlers, even for food consumers, and we look into this education thing. For example, the 5 keys and to safer foods as an example developed by WHO. If you really look into it, it's quite some common sense again, like what mommies teach the young kids. But still, if you look into it very strictly, and even for these food handlers, they are not really doing it very well. Keep it clean, toilet visiting, washing your hands, disinfection of kitchen area, separation of raw and cooked. If you try to observe how people eat hot pots or barbecue, you will see not a lot of people realize you have to separate the tools for raw foods and cooked foods. And so this is kind of knowledge still lacking from many people. Cook thoroughly, like I said, at least 70 degree is not always reached this 70 degree based on our research. Keep food at safe temperature, which is either below 5 degrees, so 4 degree, or above 60 degree. We also looked into these small details and domestic fridge temperature. Then we found, okay, a lot of people's fridge, you can measure your fridge also at home, is not as low at 4 degree, and which is falls into the dangerous zone for pathogen growth, but not for virus, of course, and virus don't grow. And also for 60 degree, and if you look into this buffet display, it's actually not really as high as 60 degree, and it's just kept warm for a long time. And in some tropical countries that I visit, you see a lot of food are just exposed at room temperature, right, at the food court. So that's also not safe practice, let's say. And lastly, safe water and raw material, as I just mentioned, for this berry, for example, especially for foods which are consumed raw. Okay, finally, I come to the end of my lecture. Now, again, still any
Yes.
question?
Yeah, still.
Yeah. Good.
Okay, I would like to ask Michael Susanto here.
Yes.
Yeah, please go ahead with your question to Dr. Dan.
Dr. Dan, may I ask questions? So, in previous slide, you have shown that we need to heat the food at 70 degrees Celsius. But is it possible that there's certain virus resistant to this heat? And can we sample by PCR? Thank you.
Do you get it, Dr. Dan?
Yes, for one thing, in general, we believe all of the viruses are not so heat resistant, let's say to 70 degree. 70 degree for around like 5 minutes, or in general regarded as safe. So heat resistance is never a problem for viruses. Also for the COVID-19, I believe if you care about it and you look into the news, people will tell you it's not heat resistant, right? Overall, not like bacteria. We are not really concerning about heat resistant over the virus. But if you say, can you tell it with the use of PCR? The answer is clearly no. Why? Because, yeah, maybe. Now see the chats. Great.
Yeah.
Okay. Why? Again, I've been talking a few times today. PCR is detecting the segments of RNA, right? So, if you treat your virus, but your RNA, specifically your target, is still there, then you cannot tell your virus is alive or not. Your virus is inactivated or not, right?
Okay, Michael, clear enough?
Can I ask again?
Okay.
So I have heard that the frozen nugget that we kept in the refrigerator, we can eat it directly without cooking. Is it healthy? Concerning about the foodborne illness.
Chicken. You eat frozen chicken directly.
No, I mean the Michael said they fry directly the chicken nugget. Do you know the chicken nugget?
Yeah.
Yeah, actually it is frozen. Then directly fry because from the frozen state.
Okay.
Is it okay? Is it safe?
I don't see any why not, because it's directly fried. No. At that temperature is no problem.
I don't mean directly fried. I mean directly eat, not without.
You eat frozen chicken.
Yes, without cooking it. Chicken nugget.
Oh, okay. That's not really. That's not really relevant for the virus, but I doubt whether it's tasty if you eat. If you really say, is it possible that it can be contaminated? Then, if yes, then it's due to foods handling, right? But overall, I don't see a correlation with foodborne virus contamination.
Okay, Michael.
Thank you.
Okay, I think we still have maybe one more question from Fauzi Ritwan. Fauzi Ritwan. Anyone? Fauzi? Are you there, Fauzi? Because Fauzi Ritwan raised hand. Fauzi Ritwan. No. Okay. Oh, this question from Sam. Okay, Sam, please. Could you unmute Sam?
Thank you. Yeah, good afternoon, Dr. Dan. In regard to COVID-19, I believe the cell is enveloped with a thick lipid membrane, if I'm correct. What stops it from being attracted to food? Thank you.
Yeah, this is actually already a question people ask me from the very beginning of this COVID-19 outbreak, let's say in January or February. At that time, I only share with you a story because there's no conclusion I can tell you. I share with you a story is at that time, people ask me, do you think people will get sick from eating buffet still? At that time, they were trying to protect, let's say, the business of buffet of these restaurants, and they say, okay, the chance should be very small, right? But then my opinion is, you know, the chance is indeed very small, but you just look into how people share the tools, how people talk to each other when they are getting food, and so on. I don't think it's not possible. That was my opinion at that time. So I say I'm conservative. I don't think it's really a big issue because it's not really the main transmission route. But if you really organize a buffet, then people talk to each other, they share a same tool, and so on. They touch each other, they touch their plates and so on. I can never say it's safe. But then afterwards, the outbreak got even worse, and I see people are getting a little bit paranoid. They are starting to talk like, you know, we detected that's news from China that you see. Okay, we detect this COVID-19 from packaging or raw meats and raw fish. Then I hear some of these people, they don't dare to buy these food anymore, and then they directly start to boil their food when they are home. That's for me, on the other hand, also not really necessary. The reason is still like what I said. I believe if you want to control COVID-19, food is definitely not a priority because this virus is not following the oral fecal routes. But if you say specifically, is there any chance that anybody actually gets infection from these foods? I can never say no. Yeah, because that's, yeah, you never know, right? But as a scientist, or let's say as the stakeholder, if you want to say I want to control this virus effectively, I believe, for example, wearing a mask to prevention people have close contact with each other is much more important than putting a lot of efforts into the food for the moment.
Thank you very much.
Okay. I think the time is almost up. So I'm not sure. Pak Aman, should we have a documentation here? Excuse me, Dr. Dan, do you mind if you activate your camera?
Okay. Yeah, it's crazy that my camera is not on top of my computer. It's,
So
yeah, so I have to do
yes,
this. Hello.
that's all right. That's all right. That's all right.
So
Okay. So I'm not sure. Paman, are we going to take the picture now for documentation or yeah? Or I have to hand to show the certificate first?
okay. Should I look into the camera now?
Yeah. Just nice.
All right.
Yeah. Okay, Paman may be taking a picture. Okay, all the. Please turn on your camera, everyone. Are you done, Pak Ahmad? Not yet?
Yes, Pak Resta.
Already done?
Finish.
Okay, finish. Okay. Okay, before I close this lecture or discussion, I have only maybe only a few of closing remarks. So the prevention in foodborne virus is the most important thing, right, Dr. Dan?
I wouldn't give that conclusion. You mean prevention?
Yes, prevention.
Yeah, definitely.
Okay.
I wouldn't say virus is more important than others. No.
So my closing remarks is the prevention is the most important thing in foodborne viruses, right?
Yes.
Okay. So actually, before we finish the lecture, then I'll try to give. We have already the S certificate. So, oh my God. I'm sorry, I lost my A certificate. Could Pak Arman share or someone? So we already prepared the certificate for you, Dr. Dan. So I think I cannot share. I already opened that certificate here. Can someone, Dr. Arman, or maybe wait. I think I can. Here you are. Dr. Dan.
Yeah, sorry. Okay.
This is our certificate of appreciation. This is
okay. Thanks.
Yeah, for delivery on foodborne viruses in the program 2020. Signed our dean and our head of department.
Would you send me the e-version?
Yeah. Someone from the
will send the.
Okay. That's conclude for our lecture today from Dr. Dev, right? Arman? Yeah? Anyone? And then we conclude, then I will close for the first session of our summer course. Okay? Dr. Tan, thank you very much for me.
Thank you.
If you want, we still have a video about overview of Indonesia. Like to watch.
I still have a meeting later.
Okay then. Very much.
You.
Okay.
See you.
Okay. At next are the overview video. First time already passed. Oh, I see.
West Papua. Pristine land in the western Pacific Ocean. ancient culture. A world of wonders, mysterious and magical. The life of a tribe in sync with nature. West Papua's archipelago. Extremely rich sea life. Adorned with emeralds of islands. Sprawling, protected in front of the coast. A heaven for divers. The richest marine biodiversity in the world. Home to majestic phantoms of the ocean. Friendliest of people. In life at its best. This is life in an amazing setting. Where Macaredo greets guests. An amazing sea adventure. The best of fish. And travel in style and comfort. Here, the ancient is always only a short trip away. Always reminding us. Life is beautiful. Amazing cultures and lovely sea. West Papua. Wonderful.
Okay, so now we will have a break. Yeah, a prayer break. After the prayer break, so everyone can break, and we start it again maybe about 3:30 p.m. Okay, in the afternoon. So anyone can turn off the video and also turn off the sound. So then please have a prayer, afternoon prayer. So we will join again in the 3:30 p.m. Okay, thank you very much. Okay. Good afternoon, everyone. Are you there yet? Anyone? Arman?
Yes.
Okay. Okay. Before we go to the next session, then I will share screen about Pak Burhanuddin CV. Or Pak Arman have the CV?
I'm sorry, Pak Resta.
Yeah.
Mr. Burhan is not joined.
Okay.
I think waiting.
Okay. Okay. While we waiting, then I will share screen about Pak Burhanuddin CV. All right.
Okay.
Okay. So our next speaker is Dr. Insinyur Burhanuddin Masud, MS. Is Associate Professor from Department of Conservation of Forests and Ecotourism, Faculty of Forestry, IPB University. He was born at West Flores at November 21 at the year 1958. This is the education from the doctoral degree, and he is the lecturer of Department of Conservation of Forests and Ecotourism, IPB University, and also the head of ex situ conservation laboratory, Department of Conservation and Forest and Ecotourism, IPB University.
I'm sorry, Pak Resta. Mr. Burhan is joined in Zoom.
Okay. I will stop share.
Hello, Mr. Burhan.
Halo, Pak Burhanuddin.
Okay.
Good afternoon, Pak Burhanuddin.
Okay, terima kasih.
Okay.
Happy to join this
Zoom
activity.
Okay.
Thank you very much.
Yeah, okay. Pak Burhan, my name is Resta, and I'm your moderator for this afternoon. Okay. So I was your former student at doctoral degree. I'm sure you remember me. Okay. And then, so maybe about this afternoon session, Pak Burhan will do the presentation first, or maybe do the presentation first and then have a discussion, or student can interrupt in the middle, Pak Burhan, if there is any question from them.
Thank you.
Okay.
Yeah.
Dan Pak Burhan, if the student have question, then I will interrupt Pak Burhan presentation. Okay, Pak Burhan?
Yeah.
Okay.
Okay.
Pak Burhan ready?
Ya, oke, siap.
Oke,
Insya Allah.
Oke, Pak. Siap.
Share bahannya dari situ atau saya?
From Bapak.
Oh, from saya. Oke.
Or wait moment, Pak. Pak Arman.
Yes.
Pak Burhanuddin already become the co-host or not?
Yes.
Oke. Iya, Pak. Dari Bapak bisa share screen.
Oh, ini.
Oke.
Try ya. Oke,
Pak. Oke. Time is yours, Pak. Silakan.
Thank you very much, Pak Reza. Thank you very much for all of you. I'd like to discuss about the wildlife ex situ conservation, wild animal captive breeding in summer course program at Department of IPTP, Animal Faculty. This is our learning objective. We talk about the concept of biodiversity, especially wildlife conservation, include about the captive breeding, as you know, as a form of a conservation program for endangered species. And the second, we talk about the introduction about wild animal captive breeding in Indonesia. And the third is about the short information about captivity of jalak Bali or Bali Mina and deer and short information about the Javan pangolin. In Indonesia, we talk about the terenggiling. Yeah. Okay, this is how to describe short information about the concept of biodiversity conservation. As we know, the conservation means save what we have, and wild animal is one of the natural resources must be conserved to save. And the other mean of conservation is wise use. And how to use the natural resource greatest good to greatest number for the longest time, yeah, or allocation of natural resources for intergeneration. Okay, next, and sort about the biological diversity means the variability among life organisms, also including inter alia, terrestrial, marine, and other aquatic ecosystem, and the ecological complexes of which they are part in class diversity with species and genetic diversity and genetic species, diversity of species and diversity of ecosystem. And the related being about the sustainable use of the wild animal or wildlife of biodiversity. And wildlife as a part of biological diversity, especially we call it wild animal. And according the three angel system, we talk about the biodiversity system, social goals, ecological goals, and economic goals. And I think it is the more important for us how to save our Earth. So because only one Earth, about ecological crisis, same multidimensional crisis. So we must do everything how to save our Earth. Yeah. So we need the new paradigm, how wise to use the species, the ecosystem, especially we talk about the wild animal. And this is how to protect the species and the ecosystem in our Earth. And as you know, ethical principles of biodiversity conservation is how we talk about the diversity of organism is good. The ultimate untimeline extinction of population and species is bad. So how to maximize our effort to protect to save the biodiversity in our Earth. Ecological complexity is good. It's the principle of ethical, and evolution is good. And the end, biodiversity has intrinsic value, and so we must make sure how to save our biodiversity. And as you know, three main activities of biodiversity conservation is protection of biodiversity, preservation of biodiversity, and sustainable use of biodiversity. It is the integrated concept of biodiversity conservation. And according to the site where the biodiversity conservation will be done, conservation are divided into in situ conservation or on-site conservation. Means the conservation in the natural habitats in protected areas like national park, nature reserve, and wildlife reserves. And the other one is ex situ conservation. It is conservation of site. Conservation outside the natural habitats and include the captive breeding. It's an important aspect of the captive breeding management is food and feeding, or the other aspect of technical management in animal breeding of livestock. And this is what describes short information about animal captive breeding in Indonesia. As you know, wild captive breeding program as a strategy of biodiversity conservation. Ex situ conservation and in situ conservation. And now we talk especially in ex situ conservation or outside natural habitat, like in zoo, animal park, animal rescue center, animal rehabilitation center, and captive breeding center. And the goals is the how to animal rescue and rehabilitation. And after we rehabilitation the species, we release to natural habitat to restore the population. So ex situ conservation mean the supporting system for in situ conservation strategy. And breeding and reproduction in ex situ conservation, how to increase population and or animal production to support sustainable use of animal to social, economic, and cultural. For food, like animal protein and medicine sources, objects or center for recreation and tourism, and use for research, technology, and science development, and conservation education to increase awareness, attitude, and skill of communities. And this is a goal or target and principle and management technical aspects of ex situ conservation, especially talk about the captive breeding center at captivity program. The goal and principle is breeding and reproduction, the wild animal or endangered species to increase population and animal production. And the second one is the how to protection and preservation of original genetic resources. And we must prepare to implementation of animal ethics and welfare. Talk about is the one principle must be about to implement in ex situ conservation or captive breeding program. And this is 7 minimum session management technical aspects about artificial habitat. We talk about case, grades of housing, food and nutrition, health and disease, breeding and reproduction, genetic controlling, product processing and using, and recording system or administration. And the key indicator of captivity successful is how to success reproduction, high animal production, and low mortality. And the second is the genetic resource preservation and protection. So we must make sure the original of genetic resource must be prepared in our captive breeding program. And the third one is how to contribute to social, economic, and cultural benefit. This is when we talk about the case study, especially captive breeding of Bali Mina and deer. This is Bali Mina, Lecopsa rothschildi. The natural distribution just only in the West Bali National Park. This is the species and categorize the conservation status is in IUCN red list as a critical endangered and appendix 1 of CITES. So it is the protect to trade in international or the global trade. This is the natural habitat, lowland forest and savanna in Bali Barat National Park. And about 10 years ago, Jala Bali were to introduce to Nusa Penida Island, beside of the Bali Barat, Bali Island, and successful to increase the population in the second habitat. We're talking about the introducing to Nusa Penida Island. So because in Nusa Penida, it is not the natural habitat or natural distribution of Bali, Jala Bali. It is a simple description about the bioecological characteristic. It is a food, fruit eater, or frugivorous, and insect eaters as the source of animal protein, like cricket, caterpillar, and breeding and reproduction in seasonal breeder, October and November, 2 to 3 eggs per nest. Minimum breeding age is 12 to 16 months, sometimes at 18 to 24 months. Highest productive age is 3 to 8 years, and maximum productive age is 15 to 20 years. It is using hole in trees for nesting. And trees for nesting, we talk about Kesambi, Scleisera olesea, or Pinada sonerata. It is the short information about biological characteristic, and based on this ecological characteristic, we transfer to how to build the captive breeding program. And this is captive breeding program of Balimina, has been conducted in 1983 through developing captive many parts of Indonesia, success in USA, Europe, and Japan. And the aim of captive breeding program is to reproduce, to increase population in order to support the conservation program in natural habitats, because 1982, the population in natural habitat just only 11 to 12 individuals in natural habitats. And so exit to conservation is the alternative program to increase the population, and after that to release the natural habitat. Captive breeding program for Balimina has been producing offspring, and release program has been successful to increase the population in natural habitat. In captivity, Balimina can mating and produce the offspring about 8 to 10 times per year per pairs, with total offspring about 16 to 20 individuals. So Balimina or Jalak Bali have changed mating behavior in captivity. In natural, we talk about 2 times only in every year, but in captivity, they can produce offspring 8 to 10 times in the for 1 year, per year. And this is Balimina that producing in captivity, breeding management, high productivity, and success in small to big product production captivity. This is a technical aspect of Balimina captivity breeding management, like artificial habitat cage, intensive management of captivity. This one is the breeding cage, mating cage, solitary cage, and incubator offspring. Yeah. So it is very, very important and successful in many captive breeding programs in Indonesia. This is a case of Balimina captive breeding in West Bali National Park. To produce the offspring, and after that, they rehabilitate and release to natural habitat. This is one. This is food and nutrition management principles based on animal habits. As frugivorous, must palatable, quantity and quality sufficient. This is an example of the food and nutrition management principles for Balimina. This one, feeding management of Balimina, some example of the food in many captive breeding program, how to use the food for Balimina in captivity. And this one is an example of types and total food given for Balimina in captivity. Vegetative, we should give banana and papaya. And animal source for as food is Hong Kong caterpillar, cricket, and egg. And artificial food concentrate, they use the fancy good food, croto crystal, and anti-stress fancy food. This is the one example of types and total food in captive breeding program of for Balimina. This is food for Jalak Bali in captivity. This is one food consumption and palatability of Balimina in captivity in our research. banana, about the 210 gram, and poor and insect ini. This is one. Okay, di sini. And okay, this is food nutrient who are given to balimina in captivity in MBOF, one of the captive bird captive bird orchid farm in Bogor. And this one, breeding and reproduction management of balimina in captivity, and very success. In this one, we talk about the house cyclus, artificial egg incubation, and after the rearing offspring and breeding cage, select for breeding program, and the other we use to sell for other captive breeding program, and use to release to in situ habitat for reinforce of natural population. This is breeding and reproduction management of balimina in captivity. This one. This is. And one point is about the rearing management of balimina offspring in captivity. Offspring are weaned artificially by animal keeper at 3 to 5 days, maximum 7 days, and after that, offspring are moved to be animal keeper from nest to incubator. Offspring are nurtured by animal keeper, feed the food protein 1 to 3 hours depending on offspring condition. So we must use intensive management to maintain the offspring in captivity. Offspring are maintained and being reared in incubator until when offspring can consume the food by itself. And afterward, offspring are moved to rearing cage until age 2 to 3 weeks. Offspring will be moved and continue to maintain in cage or will be sold depending on market demand or used for a release program to in situ habitat to increase the natural population in West Bali National Park. This is breeding management of balimina. Like this, 1 week and so 4 weeks or 1 month. Feeder have totally closing the whole of body. Yeah. And after that, he moved to release in single or individual cage to increase the growth of the body. And this one is the deer captive breeding. Information about deer species in Indonesia. Indonesia, we have Timor deer, or we talk about Rusa timorensis or Cervus timorensis, endemic species of Indonesia, and consists of 8 subspecies. This is 8 subspecies of Rusa timorensis. And the other one is we have sambar deer, Cervus unicolor, consists 2 subspecies, Cervus unicolor bronchi in Kalimantan, Borneo, and Cervus unicolor equinis in Sumatra. Sambar deer is a biggest deer in Indonesia. And the other one of Bawean deer, Axis kuhli, endemic species in Bawean Island. And the 4th is the Rusa total or spotted deer, Axis. It is exotic species from India, introduced about 200 years ago at colonialism period of Netherlands government, about 1887 years. Used as show in front of President Castel in Bogor. And until now, this is were introduced and developing in captivity and conserved in zoo or animal park in Indonesia. And now, generally we call it about the Rusa Istana. In Indonesia, we talk about the Rusa Istana or Rusa Castle in Indonesia. Okay, this is natural distribution of deer in Indonesia according to Van Bemmel. Talk about it. This is deer species in Indonesia. Rusa Timor, this one, and Rusa Sambar, and Rusa Bawean deer, and Rusa total, spotted deer, axis. Okay, this is shelter for deer in captive breeding area, artificial shelter, and natural shelter, using the big three as a shelter for Timor deer or for deer in captivity program. And we can build the artificial shelter like this. And according to food, generally all aspects of all species of deer as a grazer. Some food in natural habitat, like in Indonesia we call it about gewor, daily flower or tropical spiderwort, Camelina nudiflora, lempuyang, Panicum repens, jukut pahit atau carpet grass, Axonopus compressus, rumput teki atau nut grass, Cyperus kilinga atau Cyperus rotundus, rumput gajah elephant grass, Panicetum purpureum, paparean, Carex remota, Antanan, Cantella asiatica, and jukut jampang or Elusina indica. This is some food in natural habitat, and this is some vegetation or food for deer in the Ganjuran, about Dondoman, Rumput King, and other one. And we have tried to use some grass to give the deer in captive program. This is a vegetation of food deer, nampong, rumput teki, rumput geganjuran, and the other one, rumput gajah. And this is food for deer in captivity. Food consumption about 5 to 10% of body weight. It's generally we use this as sum to management, food management in deer in captivity. And food kits for deer in captivity: Axonopus compressus, Cantella asiatica, cassava, sweet potato as a supplement food, and Imperata cinerica. And this is one example about the food providing for deer in captivity, like livestock animal. We must build the food feeding, food area, feeding area, this one. And 5 species of grass must most palatable for Timor deer, according to Mulia research of Mulia in 1991. King grass, elephant grass, Bengala grass, Setaria grass, and paragrass. And according to Fitrianti et al., 2014, feeding the alternative food by using the silase consists of 3 kinds, and most palatable are silase or elephant grass than the others. Silase of rice straw and silase of banana stem. This is how to make alternative foods. If the from grass in natural is less, we must make the alternative food for Timor deer in captivity. This is an example of feeding management of for deer in captivity on dry season when productivity and supply of food were less or decreased. This is one. We make combination, day, morning food, and afternoon food, this one. And this is one, ransom E, B, and C. We make this one. And stimulating sexual behavior of Timor deer by using pasak bumi, tabat barito, and sanrego. It is aphrodisiac plant, and we're using how to stimulate the sexual behavior, libido sexual and sexual behavior for Timor deer. Like this one is tabat barito, stimulating sexual desire of female Timor deer using by powder tabat barito with 4 doses. And research result, not significant influence in the daily activity of sexual behavior. However, the 16,000 milligram doses can stimulate female deer estrus, so copulation happen. This one, yeah? This is our student in their research, how to use the tabat barito for stimulating sexual behavior of female of Timor deer. And we have used pasak bumi or tongkat ali, stimulating libido sexual of male timordil by using powder pasak bumi with 4 doses. And research result, not significant influence in the daily activity sexual behavior, but however, the 7,000 milligram doses can stimulate sexual of male deer to show copulation happen. And this one is Sanrego, stimulating libido sexual also. And this is not significant only that at the 7,000 milligram doses can stimulate sexual behavior. This one, processing capsule of tabat barito powder, Sanrego and pasak bumi for stimulating sexual behavior. Yeah, this one. And how using Sanrego and pasak bumi powder to stimulate sexual behavior. This one, and after use, the deer libido sexual of male deer can increase and they can happen to mating. This one, respond female timor deer after giving pasak bumi powder. Yeah, and so this is a shelter like that. And as you know, deer as an economic commodity, well-fed as medicine, testis and fetus as medicine, using attractive of deer as a tourism object, and innards, jeroan as medicine, tail as medicine, and education research. And now in Indonesia, some captive breeding program has sailed the stick of deer, stick of deer. And this one is, okay. Here it is, using the deer activity as recreation and tourism center. And last one, I will introduce 3 examples of Javan, especially Javan pangolin captive breeding program. Yes, now we do
Punten,
here.
Punten, Pak.
Yeah.
Punten, Pak Burhan.
Yeah.
Excuse me, is there any question before Pak Burhan continue with Javan pangolin captive? Is there any question regarding to? Yeah, Michael, or no?
Okay. Please.
Anyone before Pak Burhanuddin continue with Javan pangolin captive breeding program? Any question? Not yet, maybe. Okay.
Okay.
Please continue, Pak Burhan.
Okay, thank you very much. Okay, this is 3 stages of our research, and just only in Javan pangolin captive breeding program next. But now we will do in our research program about the study of behavioral adaptation in captivity and captive activity of pangolins in captive breeding cage, and the study of artificial food as a change of the natural food, and the study of breeding and reproduction technology and management. This is 3 main stages of our research, how to develop pangolin captive breeding program in IPB especially. This one in pangolin in captivity, you know, it's the female one, male, and maintaining individual in each cage generally. So adaptive in artificial habitat. And this one said the study of behavioral adaptation, and we have result our research. Pangolin has adapted in our case and in captive breeding program. This one is, and social interaction between male and female pangolin in captivity. Now, pangolin was moving and walking on the floor of cage, and this one. And stage 2, study of artificial food formulation for substitute the natural food. And now we have find the artificial food to change the natural food. As you know, natural food is very difficult and the supply is not continuous and the price is very expensive. So we must create the artificial food to change the natural food. And our research has artificial food formulas is made by utilizing several animals, especially fish, and rate of consumption is a significant effect to growth, body weight, and survivability of pangolin in captivity. And this is stage 3, study of breeding, and now in general, study on breeding and reproduction is still a challenge in the research and development of pangolin captivity. This is our challenge, and until now, we still don't know how to make a pair of the pangolin and successful breeding in captivity program. But until now, not yet. We will try again, and we hope next year, we will success to breeding and reproduction of pangolin. So pangolin, this is a special conclusion for Japan pangolins, have a good adaptive, are able to survive in captivity. One of the key factors in life pangolins in captivity has been successful provision of artificial food as an alternative to natural food supply. Javan pangolins exhibit positive social interaction as early breeding and reproduction. And implementation of Javan pangolin in reproduction technology in captivity important challenge in the captive process. So I think it is, and the alternative I will throw short information about butterfly captive breeding program. This is one shot. In our laboratory, field laboratory in IPB, we have built butterfly captive breeding program also. This one about the crocodile captive breeding. I will show just only these pictures. Sorry. And in Indonesia, is some captivity have success to breed the crocodile, especially porosus crocodile process. I think it is. Thank you very much. This is my presentation. Short information about the XT2 conservation, especially I talk about the wild animal captive breeding as a alternative how to build the wild animal, how to produce wild animal in captivity by XT2 program, conservation program. Thank you very much, Pak Resta.
Yeah, thank you very much, Pak Burhan. Okay.
Yeah.
Okay, Pak Burhan. It is. Yeah, that's all right, Pak Burhan. So, while we are waiting any question from the participants, so Pak Burhan, now we know that our animal, like what do we call it, dilindungi animal,
protected
is very. It is not possible to we have to captive, right? Because it is a highly apa namanya, dilindungi.
Expensive,
so, yeah. So is it possible? So is it possible to ask the farmer to what we call it, develop the captivity program for that species?
For example, is jalabal is the one species of endangered species, critical endangered species. Because in natural habitat, the population very, very decreased, just only 10 to 12 individuals in natural habitat. And after we built the captive breeding program, until now very, very success. And the production of offspring in outside in XT2 program has we sell very expensive. So about one pair, we can sell about 20
Million.
20 million Indonesia. Yeah, 20 million. Very expensive. But many people speed to find the the offspring, and it's very successful. Like one, for example.
Okay, so this is possible to develop the captive program from endangered species, right, Pak Burhan?
I think so, because many benefits that we can find this.
Okay. So from your presentation, I see the slide that show that we make a medicine from the horn of the deer.
Deer, yes.
Yeah.
Yes.
Is it all right to because deer is almost like endangered species, right?
You like to explore, internet. You can find the medicine from antler.
Yeah.
In
Is it all right? No,
yes.
no. Okay.
Or like to empowering the male, empowering our body. It's one. Yes.
Okay. Okay. For me, I think is there any question from someone? Come on. Time is a lot. We still have maybe around 45 minutes to the end of the session.
Yeah.
Any question?
Yes.
Michael?
Jessica, Gabriela?
Jessica?
Please.
Any question so far? Oh, we have a Benjamin Kevin. Raise hand. Okay.
Oh, okay.
Okay, Benjamin. Where are you, Benjamin? Oh, yeah.
All right. Good evening, Dr. Burhan. I'm Benjamin from IPB University.
Yes.
I want to ask, what do you think? What is your opinion on the animal that is currently in captivity program? And will they behave differently when they are finally released in the wild, such as maybe their feeding behavior or hunting behavior? And do you think that the affected behavior can be passed down onto the next offspring or the next generation of that animal? Thank you.
Yeah. Okay. Thank you very much. Good question. In our captive program, one of the goal of the captive program is how to use the offspring of the captive program produced in captive program to release in natural habitat. So before we release to natural habitat, we must rehabilitate to adaptate the natural behavior activity before we release to natural habitat. So we must make sure how to prepare the individuals before we release in natural habitat. So so far, it's a very successful. As example for the Jala Bali, is successful to release in natural habitat using the individual from captive breeding program. But before we not direct release in the natural habitat, must we meet treatment the individuals by rehabilitation, adaptation, and habituation before we release to natural habitat. And it is they can adaptive in natural habitat and successful increase the population in natural habitat. It is, but we must be careful how don't direct using the individual before we must prepare, make adaptation, habituation, and rehabilitation there before release to natural habitat. I think so. Okay.
Thank you, doctor.
Yeah.
Okay. Is there any question? Maybe from foreign student
Okay.
or from local student?
Without sacrifice. Apa? Okay. Any question?
Any question? Not yet, Pak Burhan.
Dr. Sam Amaton? Yeah.
Yes, good evening, Dr. I'm Sam from Western Sydney University. Maybe a question. The question if we can cut off the horns of deer, because you mentioned that horns can be used for medical purpose. Can the horns be cut without the deer being suffered, like without sacrificing the deer's life?
Yeah, we talk about the velvet, yeah, velvet. But in traditional, sometimes they use the antler, hard antler, just only give in hot hot water or make with other one. Sometimes they use in traditional. They use the hard antler as a medicine by using the in hard hot water or some drinking, ya. They campur itu. But for powder we use the velvet. So after hard antler is tanggal itu apa namanya leaf, and the velvet is the soft antler, and they capture and make a powder as a medicine. This one.
Okay, but that without having to sacrifice the actual deer, right? The deer. I mean, without killing the deer, in other words.
Not killing the deer. So because regularly the antler is just like our knee, we have cut and just growth again.
Yes. Okay. All right.
Yes.
Thank you.
So not kill the deer, not kill the deer. This is one breeding production, yeah? Especially like in China, in Australia, New Zealand, they just sell the velvet as a medicine.
Yeah.
In Indonesia, we have in some captive, they have sell the velvet, powder of velvet as a medicine.
Okay, thanks very much.
Thank you.
Okay. Any question?
Okay. Question? Question? Thank you.
Any question from everyone? Maybe Pak Burhan want to say something more about experience or anything about captive program?
Yes.
That not in the presentation yet.
Yeah. For example of the deer is not problem. The technically is now a very development in many area, many captive breeding program is success to breed the deer in Indonesia. And this one is we introduce for a species alternative to prospective, introduced as a livestock, new livestock for animal breeding in domestic species, like this. But until now in Indonesia, the deer is categorized as a wild animal by legal aspect. So many people push how to use the deer as a new domestication species, and we will try in our laboratory. We have developed, but not very successful at this one. And many species have success captive breeding using the experience in domestic species like chicken, and we transfer the best practices in livestock breeding to captive some bird, and very success now. And this is the new business area for many people, using the extractive product and using as a education and tourism area. It's very good. So I think it is the challenge for us how to develop and using many wild animal species to captive breeding and using our experience in the livestock or domestic species. I think it is. Thank you very much. I hope it is. I can contribute some information and stimulate our spirit to develop how to make or to build captive breeding program, especially for wild animal.
Okay, Pak Burhan. Hold. We are not finished yet because we don't have not yet taking a picture. So we're still waiting for another question. Please, anyone, please. They have a question here. The name is Pramana.
Yeah.
Pramana Ilmananda. Where is Pramana Ilmananda?
Hello, Doctor. Good afternoon.
Oh, yeah.
My name is Pramana Ilmananda from IPB University. I want to ask about a zoo as a place for conservation. Is it really an ideal place for a big animal, such as elephant or giraffe? Because zoo doesn't have a really big space. Thank you.
Okay. Thank you, Pram.
Thank you very much, Pramana. Yeah, thank you. Okay. In Indonesia, zoo is we call about the legal aspect based as a ex situ conservation institution. Yeah. They have not that mandate for business program, especially how to sell the extractive of species. Yeah. So zoo has make advantage to produce the animal and is a source of breeder for captive breeding program. So because it is make the breeding and reproduction, but not for sale in zoo, according to our legal aspect in Indonesia. They just they have tasked to produce, to increase the population, to prepare preparation, and to protect the original genetic resources, but they have not to sell. And in many zoos in Indonesia have successful to breed some wild animal like deer, like Bos javanicus, banteng gitu ya, tiger, Sumatran tiger have success breeding in some zoo in Indonesia, like so I think it is the suitable for make successful to captive the wild animal in zoo. Yeah. But one point I will relate, that's the zoo not have legal mandate for using to sell the animal they produce there. I think so.
Okay. Okay. Is it all right, Pram? Pram?
Okay, thank you, Doctor.
Okay. Any more question?
Yeah.
Okay, please, I ask everyone, please turn on your video because we are now currently taking a picture for documentation. So please, anyone, please turn on your video. Or is there any more question? Oh, Yasmin. Yasmin, raise hand, please. Yasmin.
Okay.
Yeah.
Thank you, sir, for the opportunity. So I just want to ask for captive fat breeding of exotic species. Is there some cases? Thanks.
Okay, Pak Burhan.
Do you get what I asked?
Pak Burhan, yeah. Okay, Pak Burhan, get the question? Is there any example of exotic animal captive breeding that Pak Burhan have?
Yeah. In our laboratory, we have Japan pangolin like exotic.
Yeah, exotic.
No, we have one, but in many captive breeding program in the community, they have using exotic species from outside the Indonesia, especially some species of bird in Indonesia.
Okay. Okay, Jasmine.
Okay.
Jasmine, are you okay? You right, Jasmine?
Okay, thanks for the answer.
So, is still there any question? Because after the lecture from Pak Burhan, the committee will explain about the last day of summer course tomorrow. So before we are closing, yeah, and before I give the Pak Burhan the certificate from our department, is there any question from everyone? Anyone?
Like last year, we go to visit to captive breeding area, but because now pandemic is not.
It was possible, yeah.
Not possible, not impossible.
Yeah. Yeah, I remember when I took your class that we have a visited to captive breeding, I believe in Bandung, Ciwidey.
Yeah. Oh,
yeah. We also see the deer there.
Oh. Ciwidey, yeah.
Yeah. See. Okay. Before I'm mumbling too much, and any more question? No. Anyone? Okay, 1, 2, 3. Any question?
So, who is interested to?
Oh, I see.
Try to captive breeding of wild animal like birds. Who's interested?
Okay. Pak Burhan ask a question.
Take challenge, yeah.
Yeah.
Because if we use the domestic species, is generally it's so okay. But our challenge is how to build the wild animal captive breeding. And are you interested?
Is there anyone interested in captive breeding? Oh,
yes, sir.
Muhammad Gilang. Okay, raise hand. Okay, Gilang. Go ahead, Gilang.
Yes, sir. I'm very interested for captive animal, especially deer. I think it's very interesting because it's not common, I think.
Yeah. And okay, now you can join in we are group, dear Indonesian dear community. You have joined us. Please give your HP number and handphone number, and I will recommend you to join in our we are group. Dear Indonesian dear practices and community. In Indonesia we have community. Yes. Thank you, Muhammad.
Okay. Anyone again interesting in deer or maybe bird, Bali, Balimina? Anyone? No. So this is afternoon, so maybe time to relax or sleepy or something. So Parman, the committee, Parman.
Yes.
Is it possible to close?
I think Pak Asep something.
Oh, something good. Oh, I mean to give the Pak Burhanuddin the certificate. Is it possible? Now? Parman. Oh, ini Pak Arman. Gilang already give the phone number for joining the DEAR committee.
Okay.
Okay. I think for this session for lecture from Pak Burhanuddin, I think is because no one is asking the question. I think it is. I will close your lecture session, but the summer course session is still open because Pak Asep or maybe from someone from the committee will explain about tomorrow. But before that, I will give Pak Burhanuddin the certificate. Okay, I will give you share screen first. Not yet. Okay. Okay, this is already share screen. Okay. This is our certificate of appreciation for Pak Dr. Burhanuddin Masud for delivering a lecture on wildlife ex situ conservation in the summer course program of 2020 from our Department of Animal Production and Technology, signed by the head of department and also our dean. Itu Pak, ini sertifikatnya.
Resta.
Ya.
Not yet. So, Pak Resta.
Not yet show?
Yeah.
Oh, sorry. Stop share that. Okay. Not yet. Pak Asep. Okay, Pak Asep.
Pak Asep, ya. Pak Asep.
Terima kasih.
Thank you very much.
Thank you very much, Pak Burhan. So, Pak Resta, try to show again the certificates.
Okay.
Yeah.
It's too long.
Okay.
Pak Arman, maybe from Pak Arman, if Pak Resta is not working properly.
Oh, I see.
Mas Dimas.
May I share?
Oh, yeah.
Oh, okay. Bu Yuni, go ahead.
Damn. What happened? Bu Yudi,
Yeah.
Yeah.
it's almost the same like Paresta. We could
Maybe.
not see the certificate.
Yeah,
Oh.
maybe. In my competence, it's already so, but
Oh, okay. Or maybe in my computer. I don't know. Maybe we have to ask to a student. Have you
Yes.
seen
Sorry,
the certificate?
your laptop, Asep, because in our screen is okay.
Oh yeah. Okay.
oh, okay.
Sorry.
Answer again.
Okay, Pak Burhanuddin, thank you very much for giving the lecture. Ah, I see.
Thank you.
This is the certificate.
Thank you, Dr. Burhanuddin. Thank you very much for your.
Thank you, Pak
thank you.
Dr. Burhan. Always collaborate with us. Maybe this is the third time, I think.
Amen.
Yeah.
Yes.
Thank
Happy and this opportunity.
Ya,
semoga
Allah
memberkahi. Amin. Terima kasih. Thank you very much.
Sama-sama, Pak.
Ya. Mohon maaf. Excuse me if not very maximal. I will try
Bagus,
to.
Pak. Many experience.
Yes. Thank you. Pak Resta, Pak Asep, semua, Bu
Sama-sama,
Yuni.
Pak. Mari.
Hatur nuhun, Pak. Hatur nuhun.
Hatur nuhun.
Ya, hatur nuhun. Semuanya. All of you.
Mari.
Participate.
Yuk.
Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi
wabarakatuh. Okay, Bu Yuni, please stop screen. Okay, now from the committee will be discussed for tomorrow, Pak Asep or Pak Arman? For tomorrow?
Pak Asep, something information.
Okay. Yeah. Okay, thank you, Pak Resta. So finally we finish all the lecture session, yeah, student. So we already finished 14 lectures. So we hope you can get more knowledge and experience during participant in our summer course program. Yeah. So I think a lot of knowledge and information, is very broad actually, but I think this is very important. Yeah, because we deal with animal production, we deal with food science, biotechnology, and genetic, and also animal product processing. So in this opportunity, I am a representative committee. Will explain for tomorrow. The last day of summer course program. So in previous day, we already explained about assignment. They have two type of assignment: individual assignment and also group assignment. So the first, I will explain about the assignment. Assignment. So for tomorrow, maybe I try to memorize again. So they have two type of assignment. This is Paresta. Can you see my screen?
Yeah, can.
Yes,
So
can.
this is that we already sent to your group also. This is to memorize again. So tomorrow you have to submit. You have to submit individual assignment. What kind of individual assignment is review? You make a review paper. So right here, fulfill the question, email, address, and then full name, institution, and country, date. And then you submit your document or your review to this file, yeah, and then submit it, yeah. So the deadline, yeah, of this review is tomorrow at 12 o'clock, yeah, West Indonesian time, yeah. I mean before your lunch, yeah. Hopefully you can finish your submission, yeah. Okay. So this is the first individual assignment. So sorry. So this, yeah. So I try to memorize again for individual assignment. You have to submit tomorrow at 2nd September and then 12 West Indonesian time. Yeah. Don't forget. Yeah. Otherwise we cannot give your grade and evaluation mark. Yeah. So because this is as a credit earning. Yeah. 2 credit. So we already discussed all the committee member, yeah, regarding the review paper. This is actually good information for you because our committee try to offer, yeah, you can make a review paper, yeah, try to make with the best, yeah, the best review paper because we will offer, yeah, we will offer in 3 weeks, in the 3 weeks coming, yeah. So we have an international student conference, yeah. So the review paper that we already evaluate, yeah, by a committee or reviewer, yeah, can be selected, yeah, for the presentation, yeah. I mean not only from the because we will launch this information. So the review paper that the best, yeah, according to the reviewer suggestion, yeah. So we will select it and then we will inform to you, yeah, for the presentation in international student conference, yeah. So they have a prize, maybe not only prize, yeah, but this is very good appreciation because this international events, yeah. Try to make your best, yeah, for the review paper because the review paper that the best, yeah, according to reviewer a committee will be further or continue, yeah, to make a presentation in international student conference, yeah. So it's very nice, I think. So the first stage is 2.5 million, yeah, and then 2 million and then until 6, yeah. So try to make a best, yeah. So this is the advantage for you who participate in summer course program. You are no need to submission again, yeah, because we already have your review, yeah. So but for other that who are not participant in summer course program, they have to submit, yeah. They have to submit this, for example, until 8 September, yeah. You also, you are lucky, yeah, because you participate in summer course and then you already submitted, yeah. At least you already set, yeah, your abstract or your review, yeah. So this is try, because you still have a time, yeah, until tomorrow. Try to make a good review paper, yeah. This is our message from the committee, yeah. Okay. So can you see this slide, Paresta?
I have a question.
Yeah.
Yes, I can. Yes, I can.
Yeah, Bu Irma, please.
If we submit the papers, so we also submit for international student conference, right?
Yeah.
And then we can get certificate of attendance of international student conference, right?
Yeah, yeah.
It is automatically if I submit this paper,
yeah,
in the summer course, so I also attend the international student conference. So I can get 2 certificates.
Yeah.
Yes, I can get 2 certificates. The first one is participant of summer course. The second participant of ISC, right?
Yeah.
But for the presentation in the International Student Conference, only final participants that have been selected by the committee, right?
Yeah. Yeah, it is like that. Ibu Irma is correct. So all you already submit your paper, you also have a benefit, yeah, to get the certificate from international conference as an attendant. Yeah, but if you your paper is selected, yeah, selected by our committee, at least you have. If you your paper is selected, yeah, selected by our committee, at least you have 3 paper, yeah, 3 paper. First is from the beginning, from the summer course you have a certificate, and then you also have a attendant for international student conference because you already submit your review, and you have also final, yeah, finalists, yeah, if you selected. But if no, you have 2 certificate. This like that, Bu Irma, is clear?
Okay, it's clear. It's
Yeah.
clear now.
Okay, so this is, you have, yeah, 1 work, but you have 3 benefit. Yeah, you have 3 benefit if you make a good paper. But if you are, yeah, it's not selected also is good because it is exercise, first experience for you. Yeah. Okay, so this is regarding the individual assignment, and then the second related with the presentation. Yeah, for the group. Yeah, for the group. So tomorrow, yeah, we divide at least. So try to raise hand or mention if your university is not yet included, yeah, in this schedule. So tomorrow we have at least 12, yeah, 12 group, yeah, 12 group, yeah, 12 group. First, IPB, because they have 3 group, but maybe we select 1, yeah, representative from animal production technology, and then group from China, yeah, Jiangsu. So 1 group only make a presentation, yeah, in 10 minutes. Yeah, 10 minutes is enough. Yeah, 10 minutes is enough because try to make what is very clear, yeah, and then short presentation, but is comprehensive. Yeah, okay. So maybe, yeah, 7 minutes. We will look tomorrow. Yeah, maybe after 3, and then we panel. Yeah, and then 3 and panel. Yeah, to make a remember, not to forget. Okay, University Samratulangi, and then Malaysia we make a 1 group, and then 5 University Diponegoro and UGM, and then University Brawijaya and OMM. And then after that we have I think we have a post test. Yeah, but don't think so much. Yeah, post test is only to measure before you do or you participate in summer course, and then after you participate. This is only to measure. Yeah, to measure your knowledge and experience. Yeah, it's only 10 minutes. I think Bu Yuni will prepare tomorrow. Yeah, and then group 7. So day 3 is animal product processing technology. Yeah, THT will present. Yeah, at least they have 3, but we select 1. Yeah, and then select group 8 is Australia, same. This is, and then group 9 is from Untan University Tanjung Pura, yeah, and Palangkaraya. And then from Taiwan, and then group 11 is F2 food science technology, yeah, and I think agronomy. This is, yeah, IPB University, and then group 12 University of Jember and Universitas Islam Kediri. Yeah, so this is try to note. Yeah, maybe I will share also this, but the time is we will manage again. Yeah, tomorrow at least we have 12. So we have a committee tomorrow, yeah, who will evaluate your group presentation directly. Yeah, tomorrow. Yeah, like that. Okay. Any question regarding the assignment and then what you have to do? Yeah. you have to finish 2 assignment, individual and a group assignment. Yeah, but I think you already have the time before weekend. Yeah, last weekend maybe you also try to do. Yeah. Okay. So any question regarding this topic?
Asep, I believe Adib Zainuri has a question about the review. Adib?
Hello, Mr. Asep.
Yeah.
I want to ask about the review for the personal assignment. Is it fine if we include less than 10 cited references for the review?
To include what? The reference? Can you repeat?
Because the requirement for the personal assignment is in the template is, I think it says that at least 10 or 15 references must be cited. Is it fine if we include less than 10?
Yeah, we highly recommend it because normally for review paper, they have a requirement. How many reference? Yeah. Who make more source of the publication, yeah, as a citation, it is very good, yeah, very good review like that. So that's why our committee try to decide at least minimum, yeah, 10 paper that you collect, yeah, from the publication, and then you can try to to compile, yeah, in the review. Yeah, so try to follow the guideline for the review that we already sent. Yeah, try to make a maximum. Yeah, 10 I think is very few. Yeah, if you already maybe be finished. Yeah, so
Okay, all right. Thank you, sir.
Yeah.
Okay. Pak Asep, we have another question from Andrew Chike. Chike, please go ahead, your question. Chike.
Please.
Chike. Chike.
Oh, his mic is off. Can you write in chat, Chike?
Already write it, Chike.
Oh, I can speak because I'm not in the house.
Oh, I see. So the question, sir, I have a question about group presentation.
Yeah.
You said that day 3, which means THT, animal production technology, have 3, but you only select 1. What is that mean?
Yeah. Okay, so for these 3 who have more than 1, yeah, it's better to send us, yeah, before the presentation. Yeah, before the presentation. Yeah, at least in maximum 12 o'clock. Yeah, before we will start the presentation at 1 o'clock. So try to submit, yeah, D1 and D3, yeah, at maximum the same, yeah, like individual at 12 o'clock. So a committee will decide which group, yeah, that should be make a presentation for tomorrow. Yeah, it is clear like that.
Also,
because due to the time limitation, yeah, so we have to think about it. But I mean, who are not presentation, it means the grade is almost the same for more than 1, because we will evaluate the substance, the content, and also the presentation. Yeah, like that.
Okay.
Like that, Paresta.
Okay. Any more question? Actually, I have a question about the group presentation, Pak Asep.
Yeah.
Is the topic similar with the individual or different?
No, we already explained, Paresa, about that.
Okay.
Yeah. So this is maybe I explained before.
I see.
So for the group is try to make a university consent, yeah, university origin or country or district. Try to discuss or to provide, yeah, what is the current situation, yeah, in your district, on your country, yeah, related animal genetic resources, related animal product processing, or other that we get from the lecture. Yeah, and then this is the content. Yeah, so this is make a difference by individual. Yeah, this is you can capture, for example, your interesting topic, but include in the topic. Yeah, try to make a profile. Yeah, like that. What is the current situation? Yeah.
Okay, got it.
Okay.
I'm sorry, I have another question.
Yeah.
I just want to make it clear. So we THT have three
group.
Tiga angkatan dari kami ada 3 angkatan, Pak. Nah, itu angkatan buat satu presentasi lalu diseleksi apa seperti apa ya, Pak?
You have 3 group, THT 54, 55, and 56, is correct?
Yes.
Yeah. So all you have all your 3 group submit, yeah, to that bit.ly, yeah. So for you who have a 3 group, try to submit more earlier, yeah, 12 o'clock, yeah, 12 o'clock is the maximum. And then our committee will discuss, and then we will decide who will be performed, yeah, for the presentation, like that. So it means all of your THT, yeah, have to make a presentation of the group. Yeah, it is correct. It is clear. Siapa tadi yang bertanya? Who?
Is it clear, Prab?
Yeah, thank you,
Okay.
Pak Asep.
Okay, good. Paresta, it is okay.
Any more question from the students? Sam, do you have any question? No.
Sam, yeah.
So clear now. Thank you.
Okay.
I did have a question regarding the group assignment, but now it's been explained.
For you, I think it's more happy because you representative from one continent, yeah. One continent and then one country, yeah. But try to make your best, yeah.
I'll try my best, but I'm not sure about the individual one.
Yeah. Try, okay. I think. Yeah, try.
Thank you very much.
Yeah.
Okay. From other student, Yolandani. Okay.
For the format, try to send Yolanda, yeah, in Word file, yeah, because from review sometimes we make a comment and then you have to improve it, yeah. Which one that you have to improve by the by the committee, yeah. Normally give the comment or suggestion, yeah, from your abstract or your review, like that, yeah. Try to send on Word file, yeah. Word, Microsoft, yeah.
Okay.
Word file.
Okay. Any more question? No.
Okay. So I think it's clear because they have to in hurry to make a individual and group, Paresta.
Yeah.
So try to communicate, yeah, with the group, yeah. So we can see which one is really work together, yeah, in the group, and which one maybe is individual, yeah, like that. Which group who work individual, try to do individual, but work with the group working, yeah. So try to discuss in the group, yeah, in the group level. I think like that, Paresta. So maybe I give to you again.
Okay.
So thank you very much, yeah.
Okay.
Please enjoy, yeah, for the last day tomorrow. After that, yeah, the summer course will be end, yeah, and you have also the credit, yeah, and also fully certificate for international student conference. Okay, thank you. I close. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Okay, Parman, can I close the session for today?